My study is the ARM PCR based SNP and gene expression analysis of the MTHFR allele in different type of the tumor to determine the tissue specific genetic heterogeneity. The tissue specific genetic heterogeneity is one of the most important aspects for survival for those suffering from malignancy. The genetics and epigenetics factors, these are the important aspects to explore the etiology of the cancer. Among them, the folate is one of the most important component of the MTHFR up to 30 to 40%. But in case of the rare mutation of TT, we are seen to change into completely T, the enzymatic activity decreases more than 80%, says Frost. The studies was further confirms does the MTHFR gene expression is being as a oncogenic mutations using KRAS gene? Yes. There is a differential expression you will find in a different kind of a tissue to determine the heterogeneity, and you will find in case number 5 that there is a minimum or down regulation with respect to the controls as comparison to case number 8 and 9, which is of the pancreas, the expression of the KRAS will be increased. The study was further continued of the using the stem cells markers in the same samples. The OCT is one of the most important transcription factors. When we have used in a 1D, we will not find any changes. But when we analyze the 3D analysis, we will find the lot of changes of differential expressions. But amongst them, in the case of the ovary, is showing a significant downregulation. And rest of the cases in CA breast, the increases were high. Similarly, the nanon is also the stem cell marker. We have used in all the same kind of the samples. We have found the expression of this case number 8 in the ovary, it is downregulated. SOX, because you know that there is only 3 stem cell markers, SOX, OCT, and nano. We have seen in SOX, we does not find, but the higher expression, upregulation was noticed by using 3D analysis in case of the breast cancer patients, as well as the Williams tumor. One of the most important finding of the stem cell studies in my groups, we have found the complete mutation in one case of the OCT4. Complete disappearance of find. This is one of the most important finding, which is the manuscript under preparations. Now the questions comes, how does the stem cells gene mutations maintain the pluripotency? These are the transcriptional regulations, and you will find that the OCT and nanon together regulate the expression of stem cell SOX2. And SOX2 sometimes also self-regulatory in the embryonic life. In the conclusion is the ARM PCR showing the mutation of the MTHFR alleles in the homozygous condition up to the 30% in the pancreatic tumors. The novel finding is of the OCT4 gene mutation in the pancreatic tumors, suggesting the increasing high risk factor for the development of the carcinoma due to the mutation of TT allele in homozygous conditions. Interestingly, all the tumor sampling showing the downregulation of the MTHFR expression, meaning thereby they decrease the dihydrofolate reductase, fail to convert into tetrahydrofolate enzyme with comparison to controls. And interestingly, the oncogene expression KRAS also is downregulated in case of ovary and the Williams tumor. The presently study suggests that the MTHFR gene mutation increasing high risk factors in the homozygous conditions. The stem cell gene mutation and the MTHFR variants increase risk factor in acute myeloid leukemia. I have published this finding in 2012 in Hereditary Genetics. These are my work which I have published. The MTHFR mutation increasing risk in the carcinoma of the cervix in 2013. Similarly, the MTHFR allele variants in ovarian cancers increasing high risk factor in 2014. And recently, the mutations of the rare mutation of the TTLL MTHFR studied in the pancreatic cancer in BGMMR. So this is my study which have been recently established in my laboratory, that how the MTHFR gene expression does modulate the KRAS mutations, or the stem cell mutation modulate the expression of the MTHFR, or vice versa. Thank you very much.